that involved. So I'm sort of trying to think about more like where this thing is going and how it can realize the vision. Last night I was thinking about like what to put in as my inspirational sort of slide, and I found this one. I don't know if you've seen this one. It's a really recent satellite imagery that, thanks to the German Space Agency, we basically now have this new satellite system that we can actually look at the bumps and and the lumps of the Earth. There's really, really high resolution. The way it works is like a tandem satellite system that are about like 200 meters or so, and they can basically overlay images and as they go. And when I was thinking about like biodiversity, it would be fantastic, to have a map like this, but then put the biodiversity layer on top of that. And so it's really fascinating the type of things that we now can generate with this type of remote sensing and satellite imagery. This is the Tibetan sort of Himalayan region, and this is the Appalachian mountains. And again, this is where we need to go. And in fact, on top of that, I have a back pocket too then. So we have to put it on devices like this. So you can actually look at these satellite maps now in your cell phones, but there is no biodiversity layer. So hopefully metabarcoding or methods like this is going to move into that direction, that we can also add the biological layer to these Earth observing systems. Now, what are we up against, when we think about biodiversity? Of course, you've seen the tree of life. These are eukaryotes, but on top of that, we have prokaryotes. But again, from a functional standpoint, we've been sort of categorizing these organisms into packets, clutches of course, it's going to be fantastic if we can do everything everywhere, anytime, but that's really not the, something that we can start doing. But sort of what we have done in, in various sectors is to look at things such as bioindicators, sort of groups of organisms that can give us ecological information, invasive species or pests and pathogens, endangered species, and things that we eat and put into our system, or like food and natural health products. And these are the sort of layers of biodiversity that when we think about tracking, we think something like a metabarcoding system, and there are lots of examples of these type of things in literature. So I'm not going to go into that. But what is missing really, I would say, is adding time and a space and trying to add those layers and understand then the dynamic and changes, and sort of is it some enough signal that we can deal with natural variation and then understand the man-made variation and those type of things. Metabarcoding, this is what we're up against. Workflow-wise, it's very simple, and that's good and bad in a way, depending on how you deal with it. It starts from any type of bulk material, biological material, soil, water, malaise traps, bentos, feces, and it sort of goes through a workflow that usually involves amplification of certain types of markers, in this case barcode markers, or capturing them, or you can go full-fledged metagenomics, but obviously you need a lot more sequencing for that. And typically it's done with next-gen sequencing of different types. This is Shadi Shokrullah, used to be working in my lab, and this is Mark Blackster with his big NovaSeq so depending on the scale, you can scale it, and I get into that, and then you get the data, you have reference libraries, and do bioinformatics and get the information. We try to sort of understand what are the elements of this system, and like again, we kind of naively categorize that from recovering the sequences that involves what type of sample I need to get, how many samples, where do I get them, and so on, or even the sequencing part all goes within the recovery. But these are also linked to situations like the resolution of my marker, and that is linked to the length of the marker that I need to get, and the mode of evolution. Is it a barcode marker? Is it a different marker? And then it comes to basically the annotation and analysis, all the database and coverage of the reference library and so on. These are interconnected, and an optimization needs to happen at all levels, or perhaps it's impossible to do that. With Terry Porter, Terry and I sort of tried to look at all the genomics methods that are used today and sort of are coming up for biodiversity analysis, and or vision was to see what of these methods can systematically scale up to sort of deal with the space-time elements and get us sort of what we need to build our map of biodiversity. And again, you can go and read this. This is a fairly recent paper in molecular ecology. Because we have DNA barcoding and reference libraries, I think based on our assessment, metabarcoding is that method. So it can give us, if we have good libraries especially, the type of approach that we need to track biodiversity and going forward. But reference libraries are important. And again, we talked about them, but I want to get my sort of little pitch to the mix. The fact is that we've done a great job, but we also haven't done a great job in many cases that we need them. Like we studied sort of this, all the GenBank records and BOL records, and we focused on bioindicators and endangered species. And this is the state, you know, when it comes to these type of groups of organisms, and especially on endangered species. Again, you know, these are the type of things that managers or people who really build the policies, they're really concerned. So we need to do a better job, hopefully in iBOL 2, to try to cover them. The other element is basically a standardization in both molecular and bioinformatics part, and we tried to capture this in another study that was recently published in terms of classifying these metabarcodes. These are primer pairs that are used for metabarcoding, and many of them are really not optimized for the task at hand that needs to be done and looked at. And then the amount of information that we can get, depending on what method we use for each of these primer sets, are shown here. So if we use a bootstrap cutoff, some sort of a statistical approach to try to deal with uncertainties, we vastly increase the identification certainty at different levels, you know, and again, you know, sweet spot code gene because it gives us a species level information, but I think we sort of scale at the genus level. In the rest of the presentation, I try to sort of show you some of the cases that we have been able to use this and with the scalability factor in mind. This is my famous Spanish paella that you know, I really love to show. But it's basically what we do when we go and get benthic samples, you know, freshwater bentos. And these organisms are really used very heavily by agencies, by researchers as environmental indicators, especially for aquatic system. And the way it works, we basically compare reference models. This is one of the methods, reference condition approach, with test sites, and we can basically have diagnostics through the measuring the biodiversity. The way it's been done so far, it's basically brute force, and this is, if I can get this animation to work, this is the CABIN program, Canadian Aquatic Biomonitoring Network, which has been sampling for the past 20 years or so, over 16,000 sites across Canada. This is, again, you know, brute force field work, and these are wadable rivers and streams, of course, and they accumulated quite a lot of samples, but really most of these are only sampled once, and that's how we were approached by our agency, Environment Canada, and we started thinking about what we can do with the barcoding and metabarcoding. So the way they have been doing it, we kind of call it biomonitoring 1.0, and this is looking at the, you know, a small fraction of the total sample by sampling and doing morpho identification at the level they can, usually family or genus level if you have a good field taxonomist, and it's only limited to the fall sampling, you know, for the larvae to be identified. With what we envisioned at the time, sort of we can go with biomonitoring 2.0 and get richer information. As I mentioned, genus, species level ID, and the whole sample is processed. And we started, you probably heard me talking about this project, which started 2011, but it's going on, and remarkably has been delivering really nicely for us, the Biomonitoring 2.0, which is in Wood Buffalo National Park, and adjacent to the Alberta oil sands, but also influenced by hydroelectric activities in places like British Columbia. And these are vast wetland systems that are also impacted by really variable hydrology, sometimes forest fires and so on. And imagine one of our sites is this here, and we defined sort of a sampling unit for this. And we not only did bentos, but we also sampled some of the flying insects with malaise traps, with water, soil, and sort of we kind of started getting these samples in 16 of these sites. Throughout the years, accumulated quite a lot of samples. Some of the sampling locations were sampled really heavily, more than 100, and some of them less. Multiple years, multiple markers, given that we want to get plants, protists, and also various types of CO1 primers for various types of fractions of animal fauna. So we get quite a lot of output from this project, and I'm not going to go through that. And it's been really, really a good project to be able to educate stakeholders. I'm just going to, again, focus on some of the trends that we've seen in terms of space-time with this. So I'm going to be showing a series of these NMDS plots. I'm sorry if you don't see them well, but if you can see the boundaries of these plots, that's enough here. So we have metabarcoding genus level. This is sampling at 2012. This is the morphotaxonomy of the same sites. As you can see, the DNA can separate these sites, and this is actually two seasons, June and August, so it can separate that seasonal sampling. And then 2013, still we have similar pattern, but it's not really the same. So we see some variability across the years. Now, if we go to look at all the different sites, and this here space is what we are trying to separate here, and we have genus level data. We also did the family level traits as another sort of way of organizing the barcode data, and then we have morphotaxonomy in here. And as you can see, the best result comes from the genus level to get these sites. And I summarized the trends here. These are the two systems that we have in two deltas, 4 sites here and 4 sites in this site. And this is what we see between the two deltas. There is more overlap on the morphological data that we get as compared to the DNA data. We have 55.1% of the data is shared on the DNA side. And then if you want to focus on sort of uniqueness of these, so 23% of the DNA taxa is only found in one delta versus 11% for the morphospecies. A similar pattern is seen for the other delta, in fact. Now, the other axis that we want to look at is time. So that was the space that I showed you already. And we would like to look at what's going on around, like sort of after, this is 5 years worth of sampling and doing the metabarcoding and also morphology. I should give a lot of credit for the people who did all this morphological analysis. This was enormous, and that was all done by Environment Canada. So interestingly, we will see a lot more overlap with the DNA and more separation across time. And I will sort of summarize the turnover of the community as we move, from 2012 to 2016. On top, we have the morphotaxonomy. On the bottom, we have the metabarcoding, and I will provide the sort of statistics of the difference from year to year. 2012, 2013, 31% DNA turnover, 71.2% with morphology. And then as you go around the years, the pattern continues. You see a lot more turnover with the morphology as with the DNA. So again, this was sort of unexpected for us, and we're still trying to figure out the reasons. So what did we learn from the wood buffalo work so far, at least for the bentos part, is the added taxonomic resolution from DNA metabarcoding is really key, that we can measure biodiversity at different levels in these sites. And we are sort of wondering this increased spatial, we see the increased spatial discrimination, but the temporal one doesn't follow the same trend. And in fact, the latest sort of from the morphological side of analysis seems like that there is, some more missing in the morphological data. That's how you see some of the, again, it might be because we are subsampling in the morphological side of things, but the pattern that we see temporally and spatially is sort of the opposite with the DNA and morphology. as we were doing this with Environment Canada, I think we were basically approached by another agency, Natural Resources Canada, and they wanted to do a blind test between metabarcoding and morphology. There are a lot of forestry sites in Ontario. This is the Hearst Forest. It's a very large managed forestry site, over 1 million hectares. And they have been running this project called Forest Ecological Integrity. As part of that, they're sampling watersheds within the forest, and they had 23 of these watersheds and sampled. They brought the samples, a duplicate to the lab, and then they did the morphology on their side. And then they really are concerned about the environmental factors in these managed forests in the watersheds. So that's why that's how the analysis sort of shaped up. And this is sort of trying to show the contribution of these environmental factors to each of these data matrices. In this case, we have morphological genera, metabarcoding genera, and then metabarcoding OTUs. And we're looking at total richness, everything, and then the percentage of these EPTs, the bioindicators, Ephemeroptera, Plecoptera, and Trichoptera, as well as the percentage of chironomids. And again, as we can, we sort of predicted that if you look at the only specific groups, the morphology and DNA, go sort of hand in hand. Dissolved oxygen, in both cases, is sort of the driver of the changes that we see. But if you look at the larger matrix, the morphological data has much smaller breadth, and so it links to dissolved organic carbon significantly, but the metabarcoding data doesn't have that significant role. One of the experiments that we've been doing, and we tried this with the NRCan collaboration, was to see if we can do this work just through the ethanol preservative that comes with the samples. Because again, if they want to go and look at the data through morphology or one by one specimen-based sequencing, they need the material. And if we just put them all in the blender and sequence them, we lose that voucher. So we did this with again, looking at the morphological genera and families, and the metabarcoding genera and families, and we got almost perfect match, and that was really reassuring. The fact that we can only use ethanol with moderate sequencing. If we do more sequencing, we probably can even get better results, but this was about 100,000 reads per reaction. Again, this was a sort of a journey that we're coming, and then I want to know what was the next step and how we're going to scale this up. And we were approached by the WWF Canada. WWF Canada, they've been, how much time do I have?
We've got a few more minutes, yeah.
Oh, good. So I have lots of slides. So they were doing these watershed reports. 10 minutes? That's good. And the watershed reports for WWF Canada, it's sort of the way that they can sell the information to the stakeholders and then get the environmental credit, they get basically attention. And what they found is that exactly what I showed you with cabin data, Environment Canada hasn't done enough. So their watershed reports basically is data poor, especially when it comes to biodiversity data. Their chemical and physical data, it's much richer. And so literally they came to me and said, look, we've seen what you guys have done, and we've written letters of support for your grants, so can you deliver? And this was last year. And after 6 months or so of discussions, they decided to launch this program, September 2017, but there was no really money, and we really didn't know what to do. They said, look, we get you the samples through informed and trained citizen scientists, and this is an officer of Environment Canada, this is a citizen scientist, this is a community member, and this is sort of our eDNA or Benthic sampling gear with the, that we thought, okay, this can be done, but where are we going to get the money? So at that time, Genome Canada launched a program called GAP, which sort of is to put this in the hands of the users, and really is not so much of basic research, it's sort of showing the results of the research and in a large kind of capacity building. And this is what we started to, and then we were successful in securing the funds, and that was 8 months of grant writing and so on. But we have been able to start this project since October 1st of this year. And this is going to go through, the idea is to get watersheds samples through the communities that are trained. But the key thing for us is delivering the results really quickly. And so from the time that they get the samples to the time that they get our results, ecological sort of analysis, should be less than a month. So that's the plan, and we're going forward. And again, another sort of sector that got really interested in this line of work, and I was very surprised, is the oil industry, offshore oil industry, that have been putting a lot of money into R&D in areas like engineering, ice management, radar systems, satellites, and so on, but none of them went to biology. So again, we were approached by a consortium of these R&D sort of developers that came to us, and we were able to secure some money to establish a site that is going to be doing this environmental monitoring, and that's off of the coast of Newfoundland, and most of the samples are coming from there. And as part of that, we were able to get a big toy, which I really like from the sequencing perspective. So one of the things that we've been able to start doing is that because of the ocean water and the complexity, most of the experiments we've been doing about 100,000 read, like 1 million read maximum from these samples. But the medical genomics have now converged on ultra deep sequencing, and a lot of devices are out there, including this NovaSeq from Illumina, that smallest run of a NovaSeq is equivalent of 52 of the MiSeqs. So that's really, I tried to get my head around that, and it's a lot of sequences. And if you go to the largest one, you get about 10 billion reads. These are the type of things that Naveed was talking about when mentioned human genome sequencing and so on. So again, the sequencing capacity, of course, requires samples, and that's another challenge that we need to deal with. But you can get one of the single flow cell and have multiple projects running. This is one run of this. You can do shotgun sequencing, you can do capture, you can do multiple applicon sequencing. And we have been able to really analyze only a small fraction of this from samples that we collected from water. And the results are really interesting. So if you get a sample and sequence the hell out of it with MiSeq, you very quickly, around 10 million reads, basically, or so, that's where you stop. With the NovaSeq, we see that this is exponentially increasing rapidly, and that's about 10 million read per sample. And part of it is that this is not just about the number of reads, but also the architecture of the flow cells are different, and we're still wondering. But one thing is, when you analyze these OTUs, you're getting a lot of planktons, quite a lot of algae and so on, but you don't see things such as whales or dolphins or even some of the fish species. Whereas here, those rare targets start to illuminate. And that's sort of where we have the tree of life with all those different groups that we need to monitor. I think that partly the solution might be basically going with this deep sequencing for places like the ocean, for streams or maybe soil as well. For streams and smaller systems, I think MiSeq type devices and applicon-based sequencing should be sufficient. So I foresee that a lot of the applications of metabarcoding is now going to focus on getting deep sequencing either through capture or shotgun or multiple applicons done, so that we can illuminate not only just the general biodiversity that is there, the dominant ones, but the ones that are sort of hidden and might be the targets that we have for conservation or invasive species and so on. And that's it. So I'd like to acknowledge my colleagues and collaborators, funding agencies. And Paul, I also found this for you. This is Pluto at 96, 2002, 2017. So this is the way that sort of trend sort of increased. So hopefully we'll have iBOL 1, maybe iBOL 2, and PBM having the same effect for biodiversity. And thanks for your attention.